So in the last lecture we have completed up to this. So and we thought to start from the receptors. So now we will see the different types of receptors. Okay. So what are the different types of receptors? There are mainly four types of receptors. There are mainly four types of receptors. What are they? Number one is uh, inotropic receptor, then G protein coupled receptor, then enzyme linked receptor and nuclear receptor. So what are the four types of receptor? They are inotropic receptor, G protein coupled receptor, enzyme linked receptor and nuclear receptor. Out of that, first we will study about the inotropic receptor. So in this what will happen? In inotropic receptors, this is the cell membrane. Okay. So this is the cell membrane and this is the channel. This is ion channel. Okay. So when the ligand will come and bind to the receptor, so on binding to the receptor what will happen? Opening of channel will takes place. Opening of channel will takes place. So this is the channel. This channel will open. So when the opening of channel will takes place, what will happen? The entry of positive charge or negative charge, that is charged species, will takes place. So due to which what will happen? For example, see here the entry of sodium will takes place, then or calcium will takes place, then it will cause depolarization. Why? Already, because see, when there is a cell, for example nerve cell or any types of cell membrane, inside this if positive charge is more, then it will cause depolarization and it will generate action potential. But if and negative charge more in the outside and positive charge more on the inside, then it will cause depolarization. But if it is reversed, and it will cause, and if it is reversed and it is cause only negative charge inside and positive charge outside, then it will cause repolarization, or it is called, if more negative charge are inside, then it is called hyperpolarization. So repolarization, hyperpolarization, this is called resting membrane potential also. So membrane potential, so activity of cell depends on the membrane potential. Okay. So you have to remember that in the inotropic receptor, what will happen? When the ligand will come and bind to the receptor, the activity or so opening of channel will takes place. So when the channel will open, what will happen? The ion will enter inside the cell. If the positive charge is entering or positive ion is entering inside the cell, like sodium or calcium, then it will cause depolarization. And if the negative charge is entering, it will either cause repolarization or hyperpolarization. It will either cause repolarization or hyperpolarization. Okay, am I audible? Okay, next, more second, you have to remember that the receptors are connected with the ion channels. The receptors are connected with the ion channels or ionotropic receptors. There is response produced after the activation of ionotropic receptors. Response produced after the activation of ionotropic receptors is within millisecond. Within millisecond. Okay. What are the examples of ionotropic receptor? The example of ionotropic receptor are: number one is nicotinic acetylcholine receptor, GABA receptor, 5-HT3 receptor, glycine receptor, ATP, ATP mainly P2X receptor. Okay, you have to remember one thing, that this 5-HT3 receptor, one ionotropic receptor is also there and one G protein coupled receptor is also there. We will see later. Okay. Next, number two is G protein coupled receptor. This in the G protein, why this G is coming? This G is mainly due to guanine. Okay, so in the G protein coupled receptor, the receptor is binding. It is the receptor is bonded with the G protein. This is the cell membrane. These are the ion channels. So a few minutes ago I mentioned about this. So we will see there. See here, the examples are 5-HT3 receptor, GABA receptor, and muscarinic receptor. There's G protein coupled receptor also, and see here, GABA receptor, 5-HT3 receptor, and nicotinic acetylcholine receptor. So these receptors are also binded with the ion channels, and they are also bind with the G protein. Also bind with the ion channels, and they are also bind with the guanine protein. Okay, that is G protein. So what I want to tell you is that How does this G protein binds, or how does it acts? So simply, when the drug will come, and bind to the receptor, or any type of ligand will come and bind to the receptor, the G protein will get activated. In that case, what will happen? There are some phases will be there. In first step, the ligand binding to the receptor, it will activate. So this is the ligand, this is the receptor. So when the ligand binding to the receptor, it will cause the activation. So due to which what will happen? A ligand, due to which what will happen? The G protein will be activated. See, this is the G protein. This G protein has three parts. One is alpha, one is beta, one is gamma. So this alpha will be separated from beta and gamma. Okay. So in that case, I will tell you properly. Listen. So when a ligand will come and bind to the receptor, the extravascular domain of, that is the receptor, this part is called extravascular domain. The extravascular domain of the G protein coupled receptor causing a conformational change, and due to the conformational change, that exposes a region that interacts with G protein. Ultimately, this receptor has three parts. One is external domain part, one is intermembrane part, and one is internal domain part. Okay, so there are 1 2 3 4 5 6 7. So you have to delete one. Okay, actually these G protein coupled receptor are also called serpentine receptor because they looks like snake, and they are also called 7 pass receptor because they passes 7 times. So this is the membrane, this is the cell membrane. In this membrane, this will be the external domain part, and this external domain part 1 2 3 4 5 6 7. So this passes 7 times through the membrane, that's why it is also called 7 pass receptor. 1 2 3 4 5 6 7. Okay, this part is called external domain part, this part is called internal domain part. Okay, this middle is called intermediate, and they are membrane parts. They are passing through the membrane, cell membrane, yes or no? So when the drug will come, when this drug will come and bind to the receptor, this is the drug or any kind of ligand, and it will come and bind to the receptor. What will happen? Due to the conformational changes in the receptor, the internal part such that is the interact with G protein. So this here is the G protein connected. So this G protein is gets activated. So activation of G protein will takes place. The activated G protein, that is G protein coupled receptor, interacts with the G protein. So the G protein coupled receptor interacts with the G protein, and the activated G protein, what it will do now, which consists of three subunits. It has three subunits. So it actually looks like this. Okay, this is the alpha part, this is the beta part, this is the gamma part, or this is alpha beta gamma part, like this. So it has three subunit, it will be binded there. Okay, so when this G protein will be binded like this, what will happen? The separation of alpha part will take place. The alpha subunit of the G protein exchange its bonds GDP for GTP, which activates the G protein and cause it to dissociate from the beta and gamma subunit. Ultimately, what will happen? The beta and gamma part will be there, and alpha part will be separated. This is the alpha part. So this is the beta and gamma part. So from beta and gamma part, alpha part is separated. Yes or no? Alpha part is separated. Now what will happen? The activated G alpha subunit. So this is the activated G alpha subunit. So this alpha subunit, what it will do? The alpha subunit can now interact. Yes, this one. This alpha subunit can now interact with the various downstream processing. So what are the downstream effectors? You know what is IP3, what is DAG. So IP3DAG, this is called ionocetyl triphosphate. DAG is called diacetyl glucosamine. So these are the secondary messengers. So it will activate the enzyme, and the enzyme activation will takes place. When the enzyme activation will takes place, what will happen? It will start the formation of secondary messenger. When secondary messenger will form, then what will happen? It will cause the protein phosphorylation, and when protein phosphorylation will takes place, what will happen then? It will cause the cellular effect. These protein are the different types of protein are mainly what are they are enzymes or protein or any types of biological ligands, yes or no? So due to this, what will happen? The cellular response will takes place. Wait a second, please. So excuse me. So now the duration of response is in second. In that case, see, you will see two types of effect. This is the mechanism. This mechanism is more available. So G protein part will be more, and along with that, what will happen? See, you will see the ion channel also connected with them. In that case, what will happen? Activation of ion channel will also be activated. The ion channels will activation of ion channel also takes place. So the ion channel will also be activated. It will lead to the cause, it will cause the opening of the ion channel, which will further cause either entry of sodium or calcium, or exit of chlorine or potassium, or entry of chlorine also and exit of potassium. Okay, so if these positive charged ions are entering, then it will cause depolarization, and negative charge are entering, it will cause hyperpolarization. Ultimately you will get to see the cellular effects. Okay, and this mechanism you will find, this mechanism is less. So this mechanism you will find less, but more you will find is that mechanism. Okay, so this part is G protein site. So this part is G protein site, and that side is ion channel site. So G protein site will be more and ion channel site will be less available. Okay, next, the duration of response is in seconds. The duration of response is in seconds in the case of G protein coupled receptor, whereas in the case of ion channel receptors, the duration of response is in milliseconds. Okay, response produced after the activation of G protein coupled receptor is within seconds. Just I have mentioned, it's okay. Next, what are the example of G protein coupled receptors? The example of G protein coupled receptors are GABA receptor, 5-HT receptor. What is 5-HT? It is called 5-hydroxytryptamine. So 5-HT receptor, GABA receptor, and what is GABA? It is gamma amino butyric acid. So gamma amino butyric acid receptor, GABA receptor, 5-HT receptor, and muscarinic acetylcholine receptor, and adrenergic receptors. These all are the example of G protein coupled receptors. And also you will see other examples of G protein coupled receptor are dopamine receptor, histamine receptor, opioid receptor, and ATP mainly P2Y receptor. If ATP mainly P2Y types of ATP receptors are G protein coupled receptor, can you please tell me which type of ATP receptor is P2 ion channel receptor? They are ATP P2X type receptor. ATP P2X type. Okay, so you have to remember that ATP P2X type receptor are the ion channel receptor, whereas ATP P2Y type receptor are the G protein coupled receptor. Okay, now we will see the enzyme linked receptors. So what are the enzyme linked receptors? We will see later. First, let's understand what is enzyme linked receptor. In this, the receptor will be the external domain of the receptor will be present on the cell membrane. When the ligand will come and bind to the receptor, it will be connected. The receptor is connected to enzyme. Enzyme is protein actually. The way it was G protein in the GPCR, here you will see it's an enzyme. So this will be connected to the enzyme, and what will happen? Phosphorylation of enzyme will takes place when the activation will takes place. After the activation of the enzyme, phosphorylation will takes place. Then what will happen? Protein phosphorylation will takes place. When protein phosphorylation takes place, ultimately it will act as an enzyme, or it will go here and it will cause the gene to gene synthesis. Okay, from DNA to RNA, and from RNA to gene synthesis. In this, what will happen? These things will take place inside the nucleus. In this, what will happen? From DNA to RNA, mainly takes place. What is this RNA? How many types of RNA are there? mRNA, rRNA, tRNA. This mRNA is messenger RNA, RNA is ribosomal RNA, and tRNA is transfer RNA. In that case, mainly mRNA will be synthesized. Why? Because it will take the information, okay, and further it will cause the formation of the different types of proteins or enzyme, and this formation of different types of protein and enzyme will further lead to the cellular effects. Okay, next examples. What are the example of G protein? What are the example of enzyme link receptors? The example of enzyme link receptors are tyrosine kinase, and then guanyl cyclase, and serine threonine kinase, and JAK. Did you ever get JAK-STAT pathway? JAK-STAT pathway. So this JAK is which type of receptor? JAK is a enzyme link receptor. Okay, so what will happen? The tyrosine kinase is mainly related to, it is also called insulin receptor, and guanyl cyclase is growth factor receptors. Not guanyl phosphate, guanyl cyclase. Serine cyclase is growth factor receptor. Guanyl cyclase is atrial natriuretic peptide receptor. So this is why it will mainly present on the digestive parts. Next, JAK-STAT, in the JAK-STAT, JAK is cytokine receptors. Okay, so response produced after activation of enzyme linked receptor is in minutes or hours. So in the case of ion channel receptor, it's millisecond. In the case of G protein coupled receptor, it's second, and in the case of enzyme link receptor, it's minutes to hour. Okay, next, nuclear receptor. What are the different types of nuclear receptor? The example we will see later. First, understand what is nuclear receptor. In that case, this is the cell membrane, and inside the cell, this is the nucleus. Okay, so in most of the activity will takes place inside the nucleus only, in nuclear receptor. So in that case, what will happen? The mainly hormone or different types of proteins or enzyme, and not enzyme, hormones mainly go, and it will bind to the receptor inside the nucleus. When it will go and bind the inside the nucleus, it will bind to the receptor. What will happen? It will start its activation. Due to the activation, what will happen? Gene transcription will takes place. From gene transcription, what will happen? Gene synthesis will takes place, and this gene synthesis will ultimately, what will happen? It will cause the cellular effect. Okay, so it is very longer duration. It shows response in longer duration. So response produced after the activation of the nuclear receptor is within hours or days. Within hours or days. This is most important. You have to remember. In the case of ion channel receptor, it is millisecond. In the case of G protein coupled receptor, it is second. In the case of enzyme link receptor, it is minutes to hour. And in the case of that nuclear receptor, it is hour to days. In the case of nuclear receptor, it is hour to days. Okay. Next, what are the examples of nuclear receptors? They are estrogen receptor, they are progesterone receptor, testosterone receptor, and corticosteroid receptor. This estrogen receptor, progesterone receptor, testosterone receptor, and corticosteroid receptor all are together called steroid receptor. All are example of steroid receptor. So can you please tell me what are the example of steroid receptor or steroid nuclear receptor? The example of steroid nuclear receptor are estrogen receptor, progesterone receptor, testosterone receptor, and corticosteroid receptor. The other nuclear receptors are Fursenoid receptor, retinoid receptor, thyroxine receptor, vitamin D receptors, and peroxisome proliferative activated receptor, that is PPAR. PPAR is very important. Peroxisome proliferative receptors. Okay, next we are going further about the different classification of receptors, their location, structure, and response time and effector. So ionotropic receptors are present on the cell membrane. Okay, GPCR, G protein coupled receptors are also present on the cell membrane. Enzyme link receptors are also present on the cell membrane. Nuclear receptors are present inside the nucleus. Next, their structure. So ionotropic receptor, it is pentamer. GPCR, G protein coupled receptor, is trimer. Enzyme linked receptors are monomer, and nuclear receptors are also monomer. G protein coupled receptor trimer, I explained few minutes ago, alpha, beta, gamma. Have you remember that? So this alpha, beta, gamma, like G, alpha G, beta G, gamma G. Yes or no? Next, response time. In the case of ion channel receptor or ionotropic receptor, it is millisecond. In the case of G protein coupled receptor, it is second. In the case of enzyme link receptor, the response time is minutes to hours, and in the case of nuclear receptor, the response time is hours to days. Next, effector. In the case of ionotropic receptor, the effector is mainly ion channel. In the case of G protein coupled receptor, the effector is mainly G protein. And what is this G? G is for guanine. And in the case of enzyme link receptor, the effector is mainly enzyme, and in the case of nuclear receptor, the effector is mainly DNA. Okay. Next, we will further see the signal transduction mechanism. So how the signal transduction mechanism takes place. I have mentioned earlier that after the activation of the receptor or after the activation of the enzyme, what will happen? The signal transduction mechanism will takes place. In this, a series of reaction will takes place, and after the series of reaction, what will happen? We will see the cellular activity. Okay, so this signal transduction mechanism now we are going to understand. In this, first of all, we will understand the cellular signal transduction mechanism in the case of G protein coupled receptors. Okay, so in the case of G protein coupled receptor, G is due to mainly G is for guanine, I have mentioned earlier. Okay, next, in this what will happen? The G protein consists of three subunits, that is alpha, beta and gamma. Already I have mentioned earlier. Alpha, beta and gamma are the three subunits of the G protein coupled receptor. So alpha molecule consists of GDP molecule attached to it. Alpha molecule consists of GDP molecule attached to it. Active form of G protein is called alpha GTP. Active form of G protein is alpha GTP. That's why what will happen? So this is the G protein. From this is the alpha, beta, gamma. From this alpha will always separate from the beta and gamma. Why? The reason is that this is the main active part of the G protein coupled receptor, and it will go and it will show further activities and actions. Okay. So next, enzyme activated by G protein. They are adenylyl cyclase enzyme activation. In adenylyl cyclase enzyme activation, what will happen? See, first of all, the drug or ligand will come and bind to the receptor. When it will bind to the receptor, the alpha, beta and gamma subunit are there. In this, GDP is there. So alpha adenylyl cyclase GTP. So what does it mean? It means that alpha adenylyl cyclase will cause formation, it will ultimately activate the alpha part. So after the activation of alpha part, what will happen? It will separate from the beta and gamma part. Yes or no? So what will happen further? It will cause the formation of secondary messengers. What are the secondary messengers? They are CAMP or ATP and IP3 and DAG. DAG is diacyl glucosamine. IP3 is ionocetal triphosphate. Okay, CAMP and ATP. So these are the secondary messengers. These are the secondary messengers. After the formation of secondary messengers, what will happen? This CAMP will cause the protein kinase A. So activation of protein kinase A will further lead to the phosphorylation of protein. So phosphorylation of protein will takes place, and the protein will convert into protein phosphorylation, and due to the protein phosphorylation, they are convert into phosphorylated protein, that is active protein. So after that, what will happen? Cellular effect will takes place. Okay. Next, the adenylyl cyclase. You have to remember that the adenylyl cyclase will convert into CAMP, which will further cause the protein kinase A. Okay. Cyclic AMP activation on smooth muscles. So how does that cyclic AMP will activate? So see, there are myosin and myosin phosphates. Okay, so this myosin will convert into myosin phosphate in the presence of enzyme myosin kinase. When myosin kinase will activate, it will convert myosin to myosin phosphate. Okay, and this is inhibited by CAMP. That is inhibited by CAMP. So what does CAMP do? It inhibits the myosin kinase in smooth muscles. Okay, and cause relaxation of smooth muscles. Cause relaxation of smooth muscle. So that's why CAMP is important in the case of relaxation of smooth muscle, because this CAMP inhibits the myosin kinase, which myosin kinase converts myosin to myosin phosphate. This myosin, when the myosin is converted into myosin phosphate, that is active form, what will happen? It will show the constriction of smooth muscle, or it will cause the constriction of smooth muscle. Now if CAMP causes the inhibition of myosin kinase, then what will happen? It will cause the relaxation of the smooth muscle. Okay. Now smooth muscle are, where does the smooth muscle are present? The smooth muscle are present in uterus, bladder, urinary muscle, GI smooth muscle, and sphincter muscle, iris muscle, and blood vessels. So you have to remember, where does the smooth muscle are present? The smooth muscle are mainly present in uterus, urinary bladder, ciliary muscle, smooth muscle, GI smooth muscle or GI muscle, sphincters. So where are the sphincter present? On the stomach, above the stomach, below the stomach, anterior to stomach, posterior to stomach. Then on posterior to the rectum, that is anal sphincter. Okay, and also one sphincter is present outside the urinary glands also. Okay. Next you will see iris and blood vessels. In the iris also smooth muscle will be present. In the blood vessel also smooth muscle will be present. So cyclic AMP activation on the cardiac muscles. How does it take place? The troponin is the protein. Yes or no? This troponin will be activated to troponin phosphate in the presence of enzyme troponin kinase. So troponin kinase will convert troponin into troponin phosphate. Okay. When the CMP will be present here, this CMP will activates the protein kinase or enzyme troponin kinase. When the enzyme troponin kinase will be activated, it will convert more troponin into troponin phosphate. The when troponin phosphate will be there, what it will cause? It is responsible for the contraction of the cardiac muscles. It is responsible for the contraction of the cardiac muscles. Now CMP activates the troponin kinase enzyme and cause the contraction of the cardiac muscles. CMP cause the activation of troponin kinase enzyme and it causes the contraction of the cardiac muscle. Whereas same CMP cause the inhibition of myosin kinase. Same CMP cause the inhibition of myosin kinase, and due to the inhibition of myosin kinase, the muscles, mainly the smooth muscle, will be relaxed. Okay. Now let's see. So up to this, the general pharmacology part completed. General pharmacology. After that, we will start ANS. ANS, that is autonomous nervous system. Autonomic nervous system we will start from the next lecture. Okay, thank you. General pharmacology completed up to this.